All right, everyone. I am here with Kim Branson. Kim is Senior Vice President and Global Head of Artificial Intelligence and Machine Learning at GSK. Kim, welcome to the TWiML AI podcast.
Thanks, Sam. Good to be here.
Looking forward to our conversation. Let's jump right in. You come to GSK by way of Genentech, which kind of this traditional or this historical Silicon Valley challenger. Before that, you spent a lot of time in Silicon Valley, and now you're at a large pharma. Tell us about that journey and how it came to be.
Sure. So, I guess my background is, I did my PhD in machine learning, um, really looking at applying machine learning to actually drug discovery. So, small molecule drug discovery and design was what I started off at. And this is I'm dating myself, but in the 2003 around then when where the cutting edge techniques were things like support vector machines and random forests and things like that, but doing a lot of work on doing simulation of physics simulation and things like that of small molecules binding to proteins and all the things around with that. From there I ended up doing a very sort of academic thing of spending time at Stanford and others. Did a lot of earlier work on machine learning applied there into graph convolutional networks. That sort of stuff was happening. And a lot of my time was spent actually in, I spent some time at Vertex Pharmaceuticals, but then a fair bit of time sort of in startups so things like, was involved in an early search startup that was acquired by Twitter, another company doing large scale medical records aggregation and differential privacy machine learning on that sort of stuff. And although, like all things, 90% of your time is building all this stuff to do ETLs, extract tables out of PDFs and all those wonderful things. And that was acquired by Apple. And then a lot of large scale machine learning on claims, other types of data. So all about like predicting like what's the probability of predicting disease X at time T given someone's past medical history, so you could intervene early and things like that. And from there, many friends at Genentech. I've always been involved in computational chemistry, so joined Genentech and really was, I guess, really recruited from Genentech to come to GSK, which, it's one of those things that wasn't really on my radar as a place to be. And I had some friends there, and I'd known of the company for a while, but they sort of convinced me to come in, and I realized that actually it's something very different happening here. So they brought in a new head of R&D, so Hal Barron had joined, and Howard was, worked at Calico with Daphne Cole and people like that. And I was sort of like, wow, there's a guy who really, how actually really understands what machine learning looks like and what it requires to be done properly, and sort of, really explained to me that like it's going to be such a core part of GSK, right? That like they're really serious about it. It's not just a company like I want to build a 10-person team and we'll dabble and we'll kind of half resource you, but it was going to be such a fundamental thing. So that's what sort of led me here and has led me sort of create the group and scale as we do now.
And give us some context for GSK. What's the core business and where does ML and AI fit in?
Yeah, sure. So we're obviously a pharmaceutical company. We make medicines and vaccines across a wide range of things. And so GSK it's an old company, right? It's been around for about 300 years, right? So all these companies every now and then they go through these sort of revolutions and they sort of turn themselves inside out and reposition themselves. And what was really apparent is that we have this increasing body of sort of genetic data. So these large genetic databases. So where we have lots of people, cost of sequencing has gone down. So remember, so I guess 20 years ago we had the human genome, one genome. Now we can do lots and lots of people, right? So you can sequence lots of people. You know about their medical histories, and you can basically answer a question like, here are a bunch of people that got a disease. Here are a bunch of people like Lachlan that don't get the disease, and ask the question, well, what's different in the genetics, right? The idea is that the genetics kind of points at a clue as to what you might want to do a medicine for, what's involved in that disease. So a really increasing amount of just data being generated in the genetic side of things. And the other side of things that's happening is really these technologies for functional genomics, right? So maybe the first wave you can think of was molecular biology, right? Restriction enzymes, we get plasmids, we can do genetic engineering and make a cell, make a protein. This is really the continuation of the evolution of those tools, is now with CRISPR and talons and those sorts of things, where you can actually perturb a specific gene, right? You can turn it up or turn it down in a particular cell type, and also at a single cell level. So now you've got this other set of technologies where you can start to like generate huge amounts of data at scale. And what it turns out with is that biology can measure so much more now. There's just this massive amount of measurement, and it's multimodal. So it's looking at RNA-seq on a single cell level. So that's looking at the messenger RNA made as you doing these edits. You can do cellular imaging, you can do proteomics, you've got all the omics as we call them. You got all this explosion of data. And so really you need machine learning in the middle of it to sort of make sense of the data, but also even to help you kind of make sense of all the literature you've got and able to plan sort of next experiments. And that's sort of in the discovery phase. So it's really core. We have this three-pronged strategy. That's sort of the genetics, right, and functional genomics on the side, and AIML in the middle to sort of integrate it together to help us really find the targets and design better medicines.
And so is it accurate to think of genetics as the kind of the data source there, what's happening in the gene genomics as a control point, the way you influence what's happening, and then AI is telling you what influence to exert based on what the patterns you see?
Yeah, so I think that the way I think about it is genetics gives us so this genetic databases give us a clue of what to start looking at, right? And we know it's really important because we've done studies and others have done this as well, showing that if you've got a medicine that works on a target, so remember genes encode messenger RNA encodes proteins. The proteins is the thing that does the things in our cells. And the proteins are what we would call as a target, the thing we want to modulate typically. And that might be with a small molecule, things like canonical things like penicillin or aspirin, or it could be something like an antibody, right? Which usually extracellular, not usually inside cells, and it sort of blocks something. And so the genetics is giving us a hint of what to look for, but it's not the whole story. You still need to actually it gives you a clue, but you need to sort of go and do further experiments to understand, like, well, is this the correct target, or is it something else that's in this pathway that's operating in? That's the functional genomics coming in, where the functional genomics allows us to basically, what happens when we turn this particular gene off, like lower the level of that protein? Does that look like it has the right effect, right? And it's sort of mimicking the effect of making a medicine. So we don't have to make a medicine to work out whether it works. We can use that kind of thing to inform it. So it starts to put these things together, and all these experiments now, because the cost of measurement has gone down, because we can take a single cell and make a change in a single cell, right? And then do RNA sequencing, like look at all the changes in messenger RNA on those single cell levels. It generates a whole bunch of data that's at a large scale. And that's where we sort of bring machine learning in. Maybe I can illustrate the point. One of the probably the one of the most one of the a key problem we work on is that in these large genetic databases, right? We really only know what to do with about probably 15 to 20% of what we call the variance. And a variant is where, you know, your DNA sequence will differ from mine, right? Maybe I get the disease and you don't. I've got like a particular mutation, right?
Right.
If that mutation falls into what we call the open reading frame of a gene, or the bit that encodes for the protein that gets translated in mRNA encodes the protein, we know
Hmm.
it affects the protein, and we can go and look at that protein and work out what's happening. Does it not fold, or is it fold but it's missing? It's not as active, that kind of thing.
Hmm.
A whole bunch of them fall outside of that, right? They fall into the regulatory regions of DNA. And you can imagine there's a whole bunch of control structures in DNA saying what to turn on what proteins and what conditions. And so we spend a lot of time in building machine learning models really to understand what genes those variants are regulating, right? The ones that are in the control area. All right,
Hmm.
it's not always like the closest gene. They can have quite long range effects. It might be different proteins, right? And so one thing that if we can sort of understand what things they're regulating, it gives us a whole lot of sort of potential targets to go and look for. So that's the sort of thing we use machine learning on, right? So we use it on discovering kind of things to make medicines against, so better targets, and they're genetically validated, so we know they're more likely to work. And then actually maybe things on the closer to the clinical side of the world, right? So I've got a medicine, how best to use it, who's going to respond by it, how do we measure the response, and things like that. And that's things like computational pathology or other types of things in the clinical domain.
One of the things that I heard in your explanation, this is maybe a little bit of a tangent, was suggesting that I think of variance as one of the genes gets flipped from a to a G or something like that, right? So it's you've got these four and one of them gets flipped. But I heard in your description that that's a oversimplification, and maybe not just which gene is encoded. Am I hearing that correctly?
Yeah.
Or
So our cells are sort of they're a network. We have lots of different proteins to be made, and they communicate to other proteins and form up various functions. And sometimes it's literally the amount of the protein, right, is important, right? And so there are classic diseases. You can think of these what we call a rare genetic disorder, where you've got a single mutation, right?
Hmm.
And it makes the protein functionless. A canonical example might be you think of hemoglobin in a sickle cell. You can think of factor 10A deficiencies. You don't make it effective factor 10A. You don't need to put that protein back. Some things are about like the level of the proteins it can influence the behavior. Some things are about some of these mutations means that maybe the protein doesn't get made at all. Something mean the protein gets made, but it's not as stable, so it gets turned over rapidly, so you just don't
Yeah.
have as much. And sometimes you might have a mutation that makes a protein always on. So it's always conforming its fate. So it's not regulated anymore, right? That's another thing. And because it's not regulated, it's driving the behavior of other pathways, and it leads to, you know, aberrant function, which leads to pathology and disease, right? And so it's not just as simple as like, I've got a mutant, right? What is the mutant? This mutant tells me I need to make a drug against that. It's actually, what's the effect of the mutant? And this is the kind of the thing before I was
Mm-hmm.
talking about that kind of the variant to gene problem, as we phrase it.
Okay.
The other missing piece is that kind of the gene to function problem, right? And that's that's another a key thing.
Hmm. Hmm. So you've got these data sources that come from genetics and genomics, and you're applying AI, ultimately trying to develop new drugs, new interventions. Can you talk about some of the specific use cases or problems?
Yeah, so I think that there's a lot of things. First of all, thinking about obviously taking your clues from genomics and things to come up with those targets, right? And then you have to think about, well, what's the effect of that we were just talking about, like that mutation on that target, right? So there might be, is it more stable or not? And then it might be, well, I think about how do I, what a thing is to take the target and that sort of cellular context and know that you've kind of made something better or not, right? Like, how do I know that I've actually found a good target that's moving? It's gonna become a good medicine in people, and that's where actually. So we do all of the things we build, all the models we build, actually have sort of a large sort of experimental feedback loop, right? And it's actually for example, that variant to gene model that has sort of an experimental feedback loop where we're actually doing what we call experiments as code. So we're asking the model what it needs, and it's really sort of adaptive sampling under uncertainty constraints. So, rather than having data being generated by some other process at GSK, and I'm trying to build a model of that, where I might have, you know, 900 examples a week of something I'm very good at, right? And I really like more examples of things I'm not good at. We actually sort of use a lot of sort of automated like biology. So this is biology done with robots, robotic automation to generate data and things that sort of feed back into these models. And it's the model that becomes the tool that helps us solve the problem, right? So we can use that model we built to help us map more of those variants to those genes. But then we still need to understand that gene to function file. And again, there we use automated experimentation. But in this case, we're doing things with these various cellular models. So they can be what we call induced pluripotent cells, right? These iPSC cells. So these are like stem like cells from patients that have a disease, patients that don't have a disease. And then we actually sort of want to work out, what we want to basically take the disease tissue and make it look more like the normal tissue. And when we say look like, it could be measured by a bunch of different ways. It could be measured by looking at imaging data. It could be measured looking at gene expression pattern or protein or some kind of functional consequence. And typically these assays are complicated. You can't do, with CRISPR and things like that. Sometimes you can just do what we call a genome-wide screen. I'll just do all the things, right? All 20,000 genes. These ones are so complex that you can't do that, and you sort of need to do an adaptive experimentation thing. So you can take your clues you start with from genetics and from the literature, for example, and you sort of seed a model for that, and then the model sort of makes an experiment, right? And we do everything as a sequential learning kind of problem. We do an experiment, we perturb a few genes, we look at how it moves things, we feed that data back in, and we say, okay, what have we learned? Based on that, my next best experiment is to do this other thing. So then I make another mutation. I do another sort of round of like interventions on that cellular system, and then sort of iterating. And you think about it, it's kind of like an optimization problem. I'm trying to find
Yeah, I
the
was just
best
thinking
target
that.
to move it to that, right?
Right,
And
right.
so and you want to get there with the fewest number of steps, right? And try and find the best things. And at the same time, while you're trying to make it look like, it's, you know, affecting the disease, so you have to, like, your ground truth is that cellular models, you also do other things like making sure that, like, there are certain proteins in the body that you can't touch, right? That, like, have toxicity associated with them, right? So a classic one is in, cardiotoxicity, right? Like, it's no good making something against your RA drug if it's going to give you cardiotoxic, right? So there's certain things that we know we can't hit. So there's toxicity from what we call on-target tox. If I hit this protein, but something bad happens, and then there are other sorts of toxicology. We can also at the same time have an AI system that's learning to learn which targets are toxic, which targets not to touch based on prior data and things like that. Which targets are easier to make a small molecule or not. It is a multi-objective optimization, because I can come up with targets that are really great, right? And we sit around, well, we have no idea how to make a selective medicine against that, right? So an example of one of those things is a protein involved in cancer called KRAS. It took people years to come up with a selective KRAS inhibitor. It was always a great target, but it just was really what we call intractable. So it's this optimization of finding something that moves your model of the disease in the right area, is kind of tractable, it's not toxic, right? And then we can put forward. So that's sort of the thing there. So we use machine learning in that sense to help design those experiments to carry that out.
So can we maybe take a step back? Have you talked through a specific concrete example of a project, whether you're talking about the cancer one you just mentioned, and what the data sources are, what the evaluation criteria are, and then talk through how this sequential learning idea plays out in some concrete context?
Sure. Let's talk about the variant to gene one, because I think that's something that's everybody has more of a sense of that now, especially. So what we do in that model is we have some genetic variants from standard GWAS analysis, these genetic wide association studies, and these are saying
Mhm.
these variants in this region of DNA important in the role of this disease, but we don't know which gene it is. It could be gene A, it could be gene B, it could be gene C. And so what we actually do is the system that looks at that, it treats the whole thing sort of a bit as a ranking problem, right? So top level model is like a ranking model, right? And there are a whole bunch of different features that feed into that. So think of it equivalent to web search. You're saying for this disease and this variant, what is the gene, right? And you're coming up with your rank list of genes, right? And you'd want your top ranked list gene to be first on the page, right? To be the causal gene. So
Mm.
that system actually has a bunch of different models, right, that feed into that. So one of these models are things that are like sort of these stacked encoder models that featurize off raw DNA. So they look at the raw DNA sequence and then they predict like where a transcription factor and which type may bind, whether that sequence of DNA is what we call open or closed, which is where it's packed up in open or closed chromatin. Um, there are other features that talk about whether these particular genes are expressed or not in a particular tissue type, because not all genes are turned on in all different tissue types, right?
Mhm.
Cardiac cells are different from neuronal cells, are different from skin cells. There are other features that come out of like knowledge graphs, so sort of like these node embeddings, and I can talk about how we have a very large knowledge graph we use behind things. All these different types of models, and some of them are neural networks in their own right, some of them are different types of things, are all sort of features that go into this other model. And it's again a neural network type model. But again, it's supervised in a form that we say, okay, here's a variant, and here what we think is the gold standard, gene for that variant is, right? And then we go away and you learn how to weight those particular features, right? Just much like you would train anything else. The challenge we have, right, is there isn't a massive amount of gold canonical, variant to gene features, because I just told you that we only really know what to do with 15% of them. So
Mhm.
then we have to do the experiment part. And the experiment is where we bring in the functional genomics. So what we actually do is take cell type, depending on what we're doing, it could be different types of cell types, but you can say it's a primary T cell from a human donor. We do the edit, and then we actually sequence those cells. We look at the mRNA levels, and we say, okay, we know what it was before, and we know what was afterwards. So we look at what's the differential gene expression.
Yeah.
And you say, I think this variant affects this gene. So then we go and look at that gene and look at the change in gene expression, right? And if we get it right, that gene expression falls, right? And only that particular gene, right?
Mhm.
And that becomes the training data. So then that kind of feeds back in. We rebuild the model, and off we run again. And so what it means is that the different teams that run those different sort of submodels, they also have different data sources that they will bring in, and some of them are generated from external data, some of them internal data, to build their kind of their feature factories that feed into this thing. But that's how we train the whole model. And it's it's a really interesting scenario because probably I would say 45% of the time, right, a simple model, right, which is like the variant affects the closest gene, will get you there and it'd be right.
Yeah.
Problem is, is that the rest of the time that model doesn't work, and it's not the closest gene. It could be something quite far away, right? And one third of the time from doing this experiment, it's really, really far away, and sort of not what we expected. So we've
Mhm.
been running this learning loop, which basically like builds the model and generates the training data at the same time. And as a result, the overall model that maps variants to genes gets better. And so we track that over time. So when we started off, we were mapping like there were like 15% of the unexplained things in UK Bio we could map, then we went to 24%. And now we're at 40%. So we know what to do now with 40% of our genetic variants in this database, right? And that gives us a whole bunch of new potential targets to go and explore with some of those other systems I talked about.
Got it. So is the again sequential learning loop, in some ways you describe it and it sounds like this, automated thing that's kind of continuing to iterate over time, performing an optimization across, all of the experiments you're doing. In other ways, it sounds like you're applying machine learning, it's giving you some, some features, some signals, and then it, goes into a scientist's brain that determines, what the next step is. How close is that loop?
Yeah, so the tools I'm talking about, the models we build, there are obviously scientists involved in running the experiments as code, right? So sort of like tending to the robots, depending on how complicated the experiment and the throughput we're doing, involved in that. It's really where the human scientists get involved is sort of the output of this sort of thing. Another experiment we have is involved in discovery of cancer drugs, right? And it's a concept of something called synthetic lethality, and all that means is basically all cells, most biology, we have redundant pathways of really important things, right? Tumor cells grow really rapidly, and they tend to sometimes, like, because they divide so rapidly, they can tend to like break and only have one functional copy of something. And and so what we know then is if we can identify which things are likely to break, and I can make a drug against that sort of thing, because it doesn't have a backup anymore, it selectively kills the tumor cell over your normal tissue, right? So a GSK drug like niraparib, right? It's a class of drug called a PARP inhibitor, but PARP inhibitors are involved in DNA repair, right? So basically, if you stop the DNA repair, it then basically you know acts to sort of kill the cancer cell. So what we actually do is have another system, and again, this one basically looks and tries to come up with what we call synthetic lethal pairs, right? If you see this mutation, then you can target this other particular gene, right? And so we have a common set of things we know tumor cells like mutations they get, and saying, well, what things pair with that? So again, we do experiments. We knock that thing out. We turn it up. We turn it down. We look at its effect on viability in a whole bunch of different tumor cell lines, right? But then the output of that doesn't automatically become like this is the target, go away and make an antibody against it or make a small molecule against it. That's where we interface with our experimental colleagues, because there's a whole lot of these are all narrow purpose ML systems we're building, right? So there's a whole lot of data and things like that, and things that they bring into bear to think about, to work out like how well that happens, and different experiments that they will then go and design to really, it's really a hypothesis that's suggested by this machine learning algorithm, for example. So, all the things we do are, to surface the information to work with our colleagues on this sort of thing. And really, sometimes the production of the ML model is a lot more automated, but then the use of the model is where the human scientists, right? These are tools for scientists, right? We cannot encode all the background knowledge of biology and things in the way we want. And also, the scientific literature is really messy, right? Not everything in it is correct, right? So there's certainly a role for domain expertise in that.
And did I hear you earlier reference work that you've done to apply machine learning to mine the scientific literature itself?
That's right. So one of the ways you think about it, if you're doing sequential learning and you're running all these big learning loops is like, humans, we've been doing medicine for a long time, right? We know a lot about biology and medicine and things like that. It
Yeah.
would be foolish to start from a clean slate and have to learn all that. It would take a lot more samples. So
Mhm.
there are ways to think about that, like how do I have structured priors if you're a Bayesian and things like that. So we have another group who does, you know, one of the great advances you've seen obviously has been NLP, right? So we have all these journal articles that are either in open source, like on the bioarchive, or Elsevier or PubMed, etc. Right? There's lots of those sources, and there's also data sets published as well. And so we have a group that sort of builds an NLP type model, and they're based on like BERT type architectures. Again, we're seeing encoders sort of appear everywhere.
Hmm.
And what that does is that sort of pulls out things, right? So entities, right? So it's entity and relationship extraction. So we pull out what we call a semantic triple, which is really a thing, a type of relation, and another thing, or a subject, predicate, object pairs, right? The predicates we care about are a limited set of things. And luckily, the scientific literature isn't as free form as the rest of things, people writing things, right? So you tend to say A has function and B, right? X does
Yeah.
Y, X does not do Y, right? And where X's and Y's could be genes, diseases, small molecules, you name it, when there's a set of things we're interested in. So we mine all that out. We run all this sort of thing over the literature. We pull out all those semantic triples and we stick that into a really big store. So we have a graph that's like 500 billion nodes, right? It's huge.
Is the
Now we don't
knowledge graph you
use,
were referring to earlier?
yeah. So we don't use that whole thing. What we do is we pull out things. So maybe I'm interested particularly in just genes and diseases and protein products of just genes and things like that, and I can pull out those subgraphs, and we might do some link prediction on that. So actually, it's not known, but we're pretty sure that X does do Y, or maybe there's some weak evidence for it. You can apply again. You're applying ML algorithms on top of that to build that knowledge base. And then we actually use usually node embeddings. There are different ways you think about how do I represent that data into my algorithm. It doesn't have to learn that like gene X does gene Y. We've already told it that this happens, right? So how do you represent that structured knowledge that you're confident in? And this means that we can be kind of more sample efficient, right? You view all our ML algorithms as sort of information engines. We can be more sample efficient with the data we're doing, so we learn the things we don't know, right? Rather than relearn the things we do know is a key. So that's another big area. And once you've built these knowledge graphs, there's lots of other uses you can put them to rather than just like the machine learning group, right? Plenty
Hmm.
of biologists can say, oh, well, what's new about my protein? Okay, here's all the facts known about my protein. Like, does X do Y? I've got a hypothesis that X is involved in Y, right? No one holds the scientific literature in their head anymore, right?
Yeah.
It's too complicated. So you can go and query that. But the interesting thing is, like, said before, we're a big 300-year-old company. Like, there are things that have been and gone in GSK that people have forgot, right? Like, sometimes, right? So like you actually mine your own data and go, oh wow, we did an experiment about that, or we found, here's an interesting protein someone's got. By the way, 20 years ago we worked on this, or someone worked on a related thing, and they found a molecule that affects this. It wasn't what they were looking for at the time, but we've got it on a shelf somewhere.
Hmm.
So that kind of thing becomes like the brain of GSK, and because you know it's not just scientific the papers, it's also the data sets aside of the papers. You can start to put whole data sets where people are doing these big experiments at scale and at industrial scale into these knowledge graphs as well. So there are lots of experiments where people are doing screens for a particular function or things like that, and they come up with lists of genes that are known to be involved in things. You can import that knowledge as well into the knowledge graph. So it becomes a kind of like growing reference base to use.
One thing I'm curious about is you think of kind of how your team operates against a quadrant of kind of innovating on the biology and innovating on the machine learning. I'm curious where you find them and where you want them to be.
Yeah.
In this space it's moving so quickly, oftentimes you may have to innovate on the machine learning to make it work for your application. Is that the case here?
Yeah. So I think that we have in general, a lot of things we don't work on very many things as a group. So we're about a
Okay.
120 person research group, and
Mhm.
we're quite globally distributed. I'm in San Francisco. I have people in. We have team members in Boston, Philly, London, Tel Aviv, Heidelberg, Switzerland. Right, we're kind of everywhere. Um, but a lot of things we work on, like there isn't a solution, right? There isn't a variant to gene off the shelf piece of software algorithm, right? Because the
Yeah.
data's not there. You got to build the whole thing. But there are cases where we can borrow things, and there are cases where we have to do research. So we do a lot of research into causal machine learning, because
Okay.
obviously we want to come up with things that are causal for the disease. So, like if I, and what I would say have a small level of clinical hysteresis. And all that means is basically a small change in this particular, like a drug against this thing. Like I don't have to knock it down 100%. If I knock it down 10%, has a large effect on the course of disease. That's an easier medicine to make than something like, well, if you take this down to 99% of its level in the body, then you might see a clinical effect. That's probably not a good candidate for medicine. So we do a lot of work on like causal reconstruction of causal data from network data and things like that. And there are some areas where you might start off with just taking things that have worked really effectively. So computational pathology is a great example. So pathology is when you might have seen those ready purple slides of a tumor and things like that, or a biopsy or things like that. And so typically they were like, they was their hematoxin, eosin stain slides, and they're looked at by a human pathologist who's looked at things and does things like, stage 1, 2, or 3 cancer, for example. We know there's obviously some information in the image. The higher the number, worse it is, right? Now, what happened was, we got digitization happening. So we got people started to scan these slides at high resolution, right? And these are big images, right? These are like 4 terabyte images, okay, gigapixel images. And then the other side, we've got ConvNets and UNets and things like that sort of sitting around. So the natural thing was like, well, I'll just take a UNet, I'll take a ConvNet, and I'll see what I can do. And I might want to segment things. I might count the number of types of cells on the slide rather than having a pathologist go through and do that, right? I might want to say, well, what's the tumor stroma ratio? Like, so when you take a tissue block, you might have the tumors growing here and there's normal tissue around it, right? How big is that area, for example? What are the characteristics? And so it started off with people doing those types of things, right? And those technologies work. But then almost everywhere where you go, we start to answer more advanced questions. You innovate on my methodology, right? Yeah. So some of the things we do now are, well, can I predict the genetic status of a tumor just from the image alone? Right? And you ask a pathologist there, they're like, "Oh, could you tell me whether this tumor has this particular mutation from looking at it?" They're like, "That's crazy. There's no way I can do that. I'm a human, right?" And you think, okay, well, but you can actually build models, and we've done this, that can actually predict like the genetic status of the tumor. So there are subtle micro changes in the environment and stain density and things like that due to like the changes in biological processes, right? A human eye can't obviously be sensitive to train that, but you can sometimes be retrospective and see what features has the model learned. But suddenly you're taking the ConvNets and these types of things and ResNets sort of frequently, and then you're starting to push them into different areas, and you start to tinker with them again, and then you're finding different architectures. So we're seeing that, and we see that also in cellular imaging as well, where we use the same types of things, where we're looking at cells and how their phenotypes will change, right, so what they look like pictorially, as they change when we give them a drug or don't treat them with a drug, for example. So it doesn't take much before you've taken some off-the-shelf technology, but typically you're starting with an architecture or something else like that you will then adapt to your use case, right? So that big, variant to gene algorithm, right? Well, I cast as a ranking problem. There's been lots of machine learning research into ranking problems for a long time, right? And there's off-the-shelf tooling and things like that, and ways to think about things. So those are things we start with, right? That we bring in, and but there are some things where it's wholly new algorithms and architectures and things that we're sort of having to invent as well.
And does your team publish in those areas?
Yeah. So yeah, I mean, so we publish. We publish all our code and our work, and it's kind of really important you do that, because you talked before about like how do you find people, right? So I think what's happened is there's a lot of people that have realized that there's now lots of this data appearing in biology, right? I mean, even since post-COVID, everyone's really interested in human health, right? And you want to find an environment where you have the computational resources and people to do that. But nobody wants to join somewhere and then kind of like vanish into a black hole, right? And we use the models you just talked about, like, we're only suddenly able to apply ResNets onto computational pathology and start with that because it's out there in the literature in the domain. And that's why AI has moved so fast, is that free exchange of ideas and test sets and state-of-the-art benchmarks. And we love all those things as well. So, we publish our code, right? Because it's the data that's really important, right? So, we will publish, if it's a model that we've built, we've got our code and there's a public data set and we can build a model, we would also publish that model built public data, right? Because it's the sort of the data GSK is generating allows us to build a model at much higher quality, right? That's our kind of strategic advantage, right? So it's all about the data, and then that's similar to other industries, right? Facebook publishes lots of really cool graph algorithms and things like that. They don't give you their social graph, right? Data. Similar for us, but it also means that we can contribute to the community. We're running a challenge, I think, at ICLR this year as well on gene discovery and causal discovery from networks and things like that. I think we've got 2 or 3 papers in Europe this year out of the group as well. So yeah, we publish and both in the conference proceedings and other scientific literature as well. It's very important for us.
Nice, nice. Do you think much about tooling and infrastructure platforms? I've read
Yeah,
I've read about your AI hub a bit. So I
yeah,
suspect the answer is yes, but
yeah, absolutely. So there's a few. So the infrastructure. I mean, there's one thing I learned from doing startups, like you start building infrastructure now. And the next best way to start building infrastructure is tomorrow, because it allows you to scale. And if you suddenly have infrastructure problems, it's really difficult to solve once you're in that phase. In the AI team, we have a whole group that's really our AML platform organization, and they build all the kind of tooling and infrastructure for us to kind of deal with data containers and running things and scanning algorithms and kind of those aspects. And it's about not only just GPUs, compute, and things. We do a lot of PyTorch extensively. Is our preferred platform of choice, but some of it even also comes down to the things we're looking at. We end up needing kind of like novel compute, right? So we've had a strategic partnership with a company called Cerebras, which some of you may be familiar with, right? Cerebras have one of these companies. They built like a really really amazing piece of hardware. And so we use Cerebras for a particular type of problem where we're building these encoder models on DNA. Now, what's interesting with these encoder models is we want to have a really, really large window size, right? And so you get into this, it's really challenging to build model parallel and data parallel kind of algorithms at this sort of scale. And the data sets we're passing over are really, really large as well, right? These big genomic data sets. So that was a really interesting problem that the Cerebras system is like, it's got massive throughput. It could be a really big model because of the scale of the chip, right? And the latency between that and the memory was really large. So we started working with them. We have a CS1 system. We'll have our CS2 soon, and that becomes a strategic thing that we can start to build new algorithms and play with new things on and actually build models for. And then so the compute is really important for us. It's key to be unconstrained by compute, right? To be like,
Mhm.
to think of like, what's the best way to solve the problem. We usually constrain by data, like that's the data I'd love to have, right?
Yeah,
And so this is also why we also work with NVIDIA, where we're pushing the bounds of CUDA. So we have a strategic arrangement with NVIDIA where we have people on site in London where we're making changes to low level, libDNN and things like that, or working with them on those types of things. So how do we make it easier for us to focus on like only one problem, which is the science problem, rather than two problems with like the science problem and engineering problem, right? Because the challenge we face are hard enough, right? So
mhm,
that's a key. It's a key component for us. And the other thing is like we want to think about how many iteration cycles a machine learning person can do per day, right? I don't want it to be
right.
someone sitting there like, I've got an idea, I've kicked it off. Well, I guess that'll be done in 2 days' time. I'll sit here and read the thing. I want them to be able to like look at something, have an idea, or have a bunch of ideas, kick them off, and then actually get the results back that afternoon and think about it, and then run another set. So that's also really key for us, is to be so as we grow, we've needed to add every new AML hire requires a bunch more A100s or whatever we need like added to the stack, right? There's a cost.
Yeah, you mentioned earlier you reference a feature factory. So you're
Yeah.
developing these features and you reference a feature factory. Is that a concept or an idea, or is that a physical thing like a feature store?
So you can imagine for if I've got that section of DNA and I've got my variant in it, right? There are different models that can like tell you different things about it. So the analogy would be with a web page. Well, I would have the title of the web page, the links to it, the text in those links, the content
Hmm.
of the web page, the word count, the author, the date. Those are all features about the website, right?
Yep.
And you know, the you have tools and code that could pull those things out and represent that in a featureization to some kind of ML model. Similar in this case, we look at that whole stretch of DNA and the disease it's in, the cellular context, and there are algorithms. In this case, they're models themselves that work out how to featureize that to represent that to that whole ranking algorithm. So we term those sort of things as sort of as a feature factory, right? So the ones that look at the raw DNA sequence and will say, well, this is open and closed chromatin. The other one will say, well, this gene isn't on in this cell type, right? And it might imagine the model when it's trying to learn how to rank the importance of those things to give you, its candidate list of genes, might say, well, this gene isn't even on in this cell type that's involved in this disease, so this is probably a low, unlikely thing, right? This one's in closed chromatin, that's unlikely. This one's in open chromatin, it's involved in this disease and things like that, so it becomes a good candidate. So it learns how to rank all these different things, and how to combine them. And so it's not a sort of a physical thing, but it is a sort of like it's a featureization type aspect, right? So it's basically featureization by other sort of submodels themselves.
When you think about all of the various algorithms and tools and things that kind of factor into and enable what you're doing and look forward, what are the areas that either you need the most innovation to happen in, or you're excited because you see the innovation happening, whether we're talking about algorithms or tooling infrastructure, that kind of thing.
Yeah, I think one of the things that I'm deeply interested in is robustness and reliability constraints, right? And this plays into a debate with in regulation and things like that, is that as we start to build sort of probabilistic reasoning systems, and imagine, let's take the pathology example where I have images and things like that, and we've designed it well, so we've made sure we've got a bunch of people with different backgrounds. We've got a large set of training data. We have underrepresented groups, so we've done our best case to do that. That's very important. And we built a model, and the model has good performance characteristics. Maybe 80% of the time it's correct, and it predicts someone's got gets the genetic status right, so we know then who to sequence or not. For example, I'm just using a hypothetical scenario. Um, how do we know how that model behaves when what's the adversarial image for for a pathology thing, like it's maybe an area is out of focus, right, or it's got a pen mark, or there isn't enough tumor strom. How do we know that this model fails gracefully? How can we define its bounds of operation and things like that? And for some other methods and things, it's easier to define and construct that. you know for a neural network, it's harder to know how some of those changes result in the decision boundary actually happening. And so knowing that you've trained a model that's for some of these scenarios, you would train off some performance for robustness characteristics, right? But it is hard to know how those robustness and reliability characteristics happen. So we do a lot of research in that area, and we have various groups we interact with and PhD students we sponsor. But that's a really active area that's it's not just our organization, our particular. It's across the industry and various things where people want to know those sorts of aspects, and that's where you get into monitoring and those sorts of things. But for us, it's all about knowing how I can measure that I found a good, robust solution, and it's not brittle. Right, the small changes in the input don't lead to large changes in the activation. Um, that's one key area. I think for us that looking at um, simpler transformer architectures that could lead to the same kind of performance is another really key thing, because we can train them faster and things like that. So understanding a little bit of that sort of just the you know model parameter space to performance trade-offs and those sorts of things, where you think, well, maybe there is a simpler architecture that can do just as well. That's a common area of research. And and then a lot of it actually comes down to really uh, biology is all about low end, high dimensionality, right, and sort of biases and time series, right. So a lot of our data, if you think about it, that idea where I've edited my variant in and I'm looking to see which genes change, right? Well, I can measure that 6 hours after I've made my edit, 12 hours or 24 hours, and the whole thing is changing over time, right? And so actually begin to start to increment those temporal dimensions. So this is where we have a lot of time series data, and we can generate that in biology. And this is where I think that's another area of key research. And probably the final one is really sort of multimodal, and end-to-end learning multimodal. So there, the classic example is I have some cells in a dish. I'm taking images of them, and I might pull them out and do RNA-seq. And I've also got my time series. Let's plug that in for good measure. And I want to start to look at that, and I want to build a model that can classify when a perturbation has made a cell look like the wild type cell, the healthy tissue, and made its gene expression look like that, right?
Hmm.
And I like to be able to end learn that. Right now, we typically learn the gene expression model, the cellular imaging convenet together. We maybe take the top two layers of those sorts of things, and you throw that into another model that learns to integrate them. We don't propagate the error back down through all of that just because of the complexity of the thing.
Yeah.
But that's an area where I think that, you know, could be really useful. And, typically, I mean, the convnets that we convolution size, right? That you could use attention on that. You can have these dilated and flexible convolutions where you could adapt that because it might not, picking the one that looked good for that. There could be a very specific one that could work better for that problem, right? When you want to, so that's a massive area of research for us, and because we have that in medicine, right? We have, you know, your biopsy, your pathology biopsy might be done once, right, when you're diagnosed,
Hmm.
but we can do clinical imaging every 7 or 8 weeks. So it's like a CT scan or MRI, for example. But I might do your blood work, right? I can sample, take a vial of blood, and I might look for circulating tumor DNA, right? So things like GRAIL or Freenome or those sorts of things, Garden Health, right? Those sorts of assays, we're looking at literally DNA in the blood that's come from the tumor cells, right? And sequencing that, and that could be done. They're all done at different timescales, but they're all
Yeah.
multimodal things about that particular patient, and you're trying to integrate all those together to say, what's your particular outcome going to be? Are you responding to this therapy? Where are you going to go? What therapy should we give you next, for example?
Hmm. Hmm. And to some degree, that brings us back to compute because the scale required to integrate all this together is significant.
Yeah, it's compute and data, right? Because
Yeah.
we can measure so many things, right? But we're measuring pretty often what you see in biology is we can measure more data on things that are less relevant to people. So I can learn lots of data on cells in a dish, right? But cells in a dish aren't going to give me the effect on, like, a whole person, right? I can't ever get information about whole organ failure from single cell culture of hepatocytes. But I mean, we're starting to see more complex innovations in biology, so things like organoids and things like that, where they start to have more of the complexity. And where we end up finding machine learning is actually building a bridging model from the thing that we can perturb and measure at scale, and then how well does that correlate to, to humans, where we can only really, like, we can't perturb humans. We can treat humans if we think we've got a really good thing. We can measure things about us, right? So that's sort of another area. That's one of the things that we were actually doing with this sort of this King's College collaboration that was recently in the press, where what we'll be doing is we're taking tumor samples from patients, right? And we can culture their tumor, and it's an organoid, so it's the tumor, but it's plus their immune system, and critically, it's that immune system components from that particular patient, and then we can start to see how that responds, right, with various drugs or influence, and we can measure various things about that and look at that over time. And the idea there is to sort of build a model of, how best to treat that patient. What are the characteristics, or even what is the risk? Lung cancer, for example, you might resect it. You're hoping that you've got it all and there's no secondary metastasis, but there are some people that see a higher rate of secondary metastasis than others, right? So how can you identify that, for example? So it's this really interesting interplay between the development of experimental biological techniques, the ability to generate data at scale, right? And the ability to build models to kind of connect that back to humans.
You mentioned robustness as being important, and before that, you talked a little bit about explainability. I'm curious how you think about that, and how you approach machine learning problems with those concerns in mind. Do you drive for performance and then back off to the explainability requirements? Is it the other way around? Is it some kind of hybrid?
It's a really interesting debate because a lot of the times, I think people kind of use interpretability or these types of things as a proxy for I don't trust or understand sufficiently the engineering validation, right? And so, I mean, I had the classic thing. I was like, okay, I can give you what was a simple model we like? I'm like, oh, I like a logistic regression with like 6 parameters. I'm like, okay, if I give you a logistic regression with 6 parameters, right, and maybe I'll only allow, like, positive non-zero coefficients, right? So this huge number of functional forms can be in that. Most people can't look at it in their head and really understand how that works, makes a decision, right? Or even set a threshold. So, and we use technology and systems everywhere day to day without knowing how they work, right? Like everyone in the lab. where it comes down to is actually where you want reliability constraints at how well it performs, right? And that's the sort of the trade-off. And there's a trade-off between when do we really need to have secondary checks and things like that when we're making really big decisions, right? Like you know, avionics for flying my plane, or maybe doing I'm going about to diagnose someone with something else. I really need to how do I have orthogonal sources of data? How to make sure it's robust? And there's sort of things in the discovery phase where do we need to have something that explains to a human scientist these are the key features of why we think this cell type is more like this cell type and others, and this target's pushing it in the right direction? Versus, and there's a trade-off between like, wow, we're asking the machine to do tasks that a human can't. We're pulling in so much data, and we're going to take those results and then check them in other ways, right? There, I would rather harness the full power of machine learning and not hamstring the system by saying, well, okay, here's a saliency map and having this, I agree with that or not, or the functional form. So depending on what we're doing, it's a trade-off. But what we always care about is making sure we build a robust and reliable model. So like understanding how you're assessing it, right? How you measure the performance, how do you understand that, like, you haven't somehow an information leakage and those sorts of things come through. So it's attention, but where you do find things is where you have to make sure that if you're replacing what someone would be doing manually, right? So there's a whole thing of like, well, is this thing better than me? How do I know it's working? How is a quality aspect? And usually, once you want to say is like, look, I'm here to automate the boring, right? So you can actually do and go to high level science, then spend your time, looking and analyzing this, right? And also giving them sort of an audit trail. They can go back and look at the data that went into the system and maybe look at it from itself, or diagnosis tools, right, on the model's performance as well. So things like, is the input vector within a vector space that's well bounded by the training set and the test set? What does the error manifold look over that thing? Is it uniform? Are there spiky regions where because a lot of our performance measures are global measures of model performance, but you could easily take the input vectors, you can tile them on a 2D plane, you can work out the error function over those things, right? And you can say, oh wow, overall it's a pretty good model, but like, small molecules that look like this thing's lousy at, right? So
Mhm.
having uncertainty bounds, those sorts of things they go a long way to actually putting these things in production. And another thing is that it's really important not just to have any model ever spit out a number. Anything we put in production has to give you both a number and a confidence bound, and also has to refuse to return a value. They're saying, I don't have
Mm.
that. This is
Mhm.
so far out what I've seen, boss. I have no idea. We can collect those, we can log those. Maybe we've got enough of them, we can build another model. And over time, almost every model becomes a cascade function. It's like, well, this is a global model for this one, this is a model for this. Maybe
Mhm.
eventually we can unify them again. But that's really important. Those are all the sort of the functional things, because it's honestly, it's quick to make a model. And once you give people a tool, and it's very quick for them to use it. But it's the opportunity cost of the downstream decisions that they can make with it, right? They decide to do experiment A and not experiment B, for example. So we think a lot about those sorts of things. And when you start off, it's always, I want to know how it works, what's the model thinking, and things like that.
Yeah.
But a lot of those theories of mind are not really truly how the model's thinking. Even if we do
Mhm.
distillation, right, and things like that, that's
Mhm.
not really it is. It's sort of a hack on those things.
Getting models to predict or describe their confidence, that's an active research area itself. Does that requirement that your model spit that out, does that put a limit on the types of architectures you use, or is it itself kind of a area where you have to question reliability and trust of that confidence element?
We do a lot of work on that internally. I think I guess Francesco Farina's got some pretty good papers coming out on that he's published that we've done on that sort of aspect. Um, we try and have some generic methods we can bolt onto any architecture, so you can kind of separate the problem a little bit. Some architectures also can give you at the same time estimates of that, depending on what you're doing. But we also, depending where we are in development and where we're using it, you can you might have greater or less requirements for that kind of aspect of things, right? So there's a bit of a certainly once you're making big important decisions and you're putting things in production and it's going into other processes, you definitely need to have those aspects. But I wouldn't say every model built at GSK by every group will always have those characteristics. For us, where we're bringing these big things that have big downstream consequences and decisions, you need to have that sort of place. And I think that you know I think just in the community globally, people are realizing that, right? you know, and that's also about sort of monitoring things in production, right? You know, of checking things, and there's plenty of examples of models going rogue and that kind of thing. And then so for us, it's a key thing, but we, depending on the stage, is and how stringent we are on those requirements for it.
Maybe kind of one more direction to briefly explore, kind of zooming out. The Can you speak a little bit about the you know kind of building an organization like yours in the context of a large company, large pharmaceutical company, transformation implications, organizational receptivity to probabilistic models, that kind of thing? Is it, it's a research organization or a scientific organization at its core. So do you, not have the resistance that, other places have, or?
Yeah, look, I mean, I wouldn't have come here if it wasn't for having, like, Hal as the head of R&D, because you've got someone who really gets it and knows. You got to go, all right, Pyth himself, like he's an engineer as well. Um, it's really important to have an organization that, because it's core to the strategy, like people are like, oh, we're going to have AIML, we're going to do it. And sort of when I came in, I'm like, look, we get like the normal way you hire and do people doesn't work anymore, right? I want to interview people and I want to give them an offer in a few days, like within like 3 days. We're going to do it. We're going to use HackerRank. We have all these different things we bring people in, and we're not going to do the usual process. So they get an offer in 3 months' time or a month, because they've got other places to be, right?
Mhm.
We want to show them that we're not this giant ossified organization, that we can move fast and do things, right? Um, even to the way we're working, we can use Slack. We're going to be distributed everywhere. We're going to go where talent is. We can use a laptop and a thing. We can work that way.
Mhm.
Post COVID, I think the rest of the organization's caught up to that. So we came in and did things in a very, very different way, right? And like from the way we interview, HR, hiring, but also we all use Macs, like this HPC requirements, how we're going to do things, and so we built like a whole new processes to do things, right? That are the way we work, right? We work in 2 week sprints. We have these different types of things. What's interesting is actually seeing the wider organization kind of being given permission to think and innovate, and like some of them picking up some of those tactics and things like that, and the methods of managing science, right? Because I first talked to my biological colleagues, they're like, you understand, biology doesn't work that way. We can't plan things out for like this could take longer or shorter. I'm like, well, computer science doesn't work that way either, right? Like we actually like, it's I love to think that like we know the thing and we write down all the steps and we just do it. That's not how it works, right? So it's always a garden of forking paths. So doing that, stopping every 2 weeks, going what worked, what didn't work. Okay, now we'll do this. Lets you have a really good way of planning and working across really complex teams as we do. So we've brought in a lot of that culture. There are always people that are true believers, right? That are like, oh my gosh, ML and AI can do everything. You're like, whoa, slow down. Like, let's just talk about what we're doing here. And then there are people that are super skeptical, right?
Mhm.
That are like, well, how is this anything different from before? This sort of stuff, or to the, no, you'll never replace a human scientist, human ingenuity. I've got a better way of picking things. I can synthesize this on my head. And maybe they really do have an alpha value. Maybe they really do have that. But sometimes a lot of them, I'm like, yeah, I think you got a fair bit of recall bias there as well. So it's a tension and a trade off.
Mhm.
And like all these things, we'll work out where some of these tools, based on the technology maturity stack, where they're best used, and where we're too early, right? Um, or where we don't have enough data or the right data, that sort of thing. But it's been fun, and it's been fun to sort of as the organization's grown and more people have come in. And I think what people have realized is that if you're interested in doing machine learning in bio and these sorts of things, that um, companies like GSK actually have lots of compute. So if people that like Jeremy with MIT, it's like now I've got more compute and more data than I ever had, right?
Mhm.
And I don't have to write grants and spend all my time doing these sorts of things. And if you get it right, you've got a whole machine that will translate to an impact, right, to patients, to really do those sorts of things. And that's really important to a lot of people as well, is that connection to the part of the whole thing, rather than, okay, I'm an academic, I built some good idea, now I have to make a startup and do the whole thing and it's going to take like so long for my work to get out there and actually influence the world. So those are all good things. There are certain pluses and minuses to doing things in large corporations to smaller ones, right? A small
Mm.
startup, we can raise capital, we're all in the same room, or place, we know what we're doing, we're all in charge, let's go. We don't have to have all those overhead big things. Large corporation takes some time to turn the ship, but once you can focus that whole thing on something, man, you can really drive on it. So different skills. Certainly the largest corporation sort of thing I've sort of worked in, and but the organization has to want to do it. I think is
Mhm.
the lesson I've learned. Like, if they're not really into it, or the senior leadership aren't really into it, or a large fraction don't, it's not a core strategy. It's a very difficult thing, right?
Mhm.
And different companies are in different stages, right? Some of them are externalizing it, but we decided to build a really large in-house team.
What are the couple three top three things that keep you up at night? Like, what do you most worry about in your role?
The first thing is being able to generate the data at the right kind of cadence, right? So one of the great, if you're in different domains, right, you can, depending on what you're doing, you can get, high frequency, lots of new data generated quickly. For what we have to do, for example, if you think of like reinforcement learning, right?
Mhm.
I've got a simulator of the game or things like that. I can get many, many samples. I can run lots of experiments, right? I'm in bioland, right? Like, I don't
Yeah.
get, 12 million data points. I get like 300 data points every 4 to 6 weeks, and they
Mm.
cost. And by the way, it costs us a lot of money to generate those data points. So then you start to ask this question of like, what's my information gain? What's my model performance gain per data point per time? And where am I? Am I linear? Am I plateauing? Like, how many cycles do I need to run? I can run 12 cycles a year. I can get so many data points. Is that enough? Right.
Mm.
So it's all about, for me, a lot of it is about can I, am I ever going to be able to generate enough data to solve this problem, or generate the right data? Um, and another thing is that the cost of the experiment. But people will ask like, well, how much data do you need to build this model, Kim? And I'm like, I don't really know yet. You know, I need more, but then we'll start to see a trend. But or am I collecting the right data? So it's really about those learning cycles and those sorts of aspects. I'm I'm really, the other thing that sort of keeps me up enough is thinking about the best ways and the ways that we have other data sources and things that we generate data. There's lots of historical data in GSK that we pull it together and use it in the right fashion, right? And that we remove. It's really important to these are patient data, it's about individual people, right? But when we run a trial and we do things for people, they're contributing to medical research. And if you talk to a lot of people in trials, they're like, you say, well, we ran your trial and we put it into a box and the medicine was successful or not, right? And like no one else can touch that data, right? I'm like, that's criminal, right? They contributed medicine. There's other things we can learn about it. So what I'm concerned about is when we generate data and we're doing things as an organization, how do we make stackable data sources to build this like longitudinal corpus, right? An individual medicine for a particular, for say rheumatoid arthritis, it may fail, right? Which is terrible for patients and terrible for us. Our drug didn't work as well as we hoped. But the question is, what do we learn from it? And how do we build data sets that have the same common longitudinal characteristics, maybe a common cause? I can join them up together and I can build this longitudinal corpus of data. And so a lot of the time when people are doing an experiment in the lab, they do an experiment, and then, they'll analyze it for that particular use case, and then we lose the metadata or it's lost and things like that. So I like to tell people like, build data for future you so you can use it again. And also like, collect those other data points at additional marginal cost, right? Like that are really useful. So those are the things that really keep me up. And the final thing is really about like, if I'm building models in pathology, we're starting to do things that are really like, doing patient prognosis, doing prediction, doing these sorts of things, saying this person's likely to benefit from this medicine or not. As we start to go into and learn that, is to like, we have to be right. And the other thing is we make medicines for everybody. The challenge we have is if we're using lots of prior information, or I'm relying on a system where I can get digitized medical records or pathology can be uploaded. We can raise the risk of building really great AI advances, but only work for people that are in countries that have the data infrastructure and things like for that. So we think a lot about what are the data, what we call, like, what's the data cold chain? We have a cold chain for vaccines and medicine. What's the equivalent of that? We don't want to build these great advances and say, oh, I'm sorry, that's it's going to take 5 to 10 years for another country to be able to have access to it, right? Those are my top three things.
Well, Kim, thanks so much for joining us and taking the time to share a bit about what you're working on and how you think about the problems in your space.
Thanks, Sam. It's been a lot of fun. I'm a big fan of the pod. So cheers. Thank
Thanks
you.
so much. Thanks so much.